Okay, so we talked about energy and enzymes and its application in photosynthesis, and now we're going to switch gears and kind of talk about a similar but opposite reaction. It's not completely opposite, but some of the processes and end products and beginning products are the opposite of photosynthesis. And this takes place in mostly in the mitochondria. Some of it takes place in the cytoplasm though. So the process we're talking about is cellular respiration. And this is the process of converting organic molecules, sugar, amino acids, and lipids, and using oxygen and converting it into energy. ATP being the energy molecule we use. So autotrophs are organisms that synthesize organic molecules through the process of photosynthesis, which we talked about last time. And then most other organisms, all organisms that aren't autotrophs are heterotrophs, where they have to eat other organisms and break down the potential energy in the structures built by autotrophs for their metabolic processes. And cellular respiration, similar to photosynthesis, is a series of reactions starting with organic molecules to create a proton gradient, again just like photosynthesis, which is then used to make ATP. Redox reactions are vital for this, and if you remember, redox reactions include oxidation and reduction. In reduction, you gain an electron or proton, and in an oxidation, you lose an electron or proton. Dehydrogenation is a reaction where the lost electrons are accompanied by protons. And so you have oxidation, reduction, and dehydration reactions occurring. So one of the reactions which is prevalent in cellular respiration is the reduction of NAD+, okay, which is a similar molecule to NADP in photosynthesis. And it gains a proton and 2 electrons to become NADH. The opposite of that is the oxidation of NADH, which donates a proton and 2 electrons to become NAD+. So respiration then is a sequence of these redox reactions. The first is the oxidation of organic compounds. The second is the reduction of electron acceptors, NAD plus and FAD plus, to NADH to FADH2. So what you're doing is building potential energy here through the oxidation of organic compounds. And then you move those to be reduced again, to be oxidized again, and then reduced by oxygen. And oxygen then becomes water. So we're going to break this down into its smaller components. Oxygen is the final electron acceptor in aerobic respiration, and that's why we breathe in oxygen. There are other molecules besides oxygen that are used in anaerobic respiration, and these are done by bacteria that live in hydrothermal vents or in geysers, where they have there are chemical reactions occurring there, where they can use something else, some other molecules, as the electron acceptor. Fermentation then is the electron acceptor is an organic molecule, and we also go through fermentation, but it's more efficient to do cellular respiration. So in aerobic respiration, one glucose molecule has enough free energy, has 686 kilocalories per mole of glucose. Okay, that's the free energy. And it can be even higher than this in a cell, in an enclosed environment. And this large amount of energy has to be released in small steps to gain as much energy out of it as possible. So here is our equation for cellular respiration. Sugar plus oxygen makes carbon dioxide and water. But it has multiple steps. Again, this is an oversimplification. There are enzymes and many other molecules involved in the process. So again, ATP is the energy molecule, which has 7.3 kilocalories per mole of free energy. And there's two mechanisms for synthesis. One is the substrate level phosphorylation, and this is where you transfer phosphate group directly to ADP. Happens during a process called glycolysis, which we'll go over in more detail. The second one is oxidative phosphorylation, which uses ATP synthase, an enzyme which we have run into before, and energy from a proton gradient to make ATP as the proton passes through from higher to lower concentration. Alright, so the complete oxidation of glucose proceeds in 4 stages. The first is glycolysis, so where you break down glucose. Then you have pyruvate oxidation, the Krebs cycle, and the electron transport chain and chemiosmosis. Okay? Glycolysis is the breaking down of glucose into 2 pyruvate molecules, and pyruvate is a 3-carbon molecule. Okay, which makes sense. You start with 6, split it up, it becomes 2 3-carbon molecules. This occurs in the cytoplasm, and you have a net production of 2 ATP molecules, so you get 2 energy molecules out of this reaction. But it also produces 2 NADHs by reducing 2 molecules of NAD+. For glycolysis to continue, you have to have NADH. You have to have a supply of NADH. Okay, so one way you can resupply NADH is to, you don't need NADH, you need NAD+, right? And so one way you can do it is have NADH sent off to be used for aerobic respiration, or you can have it converted back to NAD+ in fermentation, okay? Which would be up here without oxygen. You go through fermentation, you can reduce it there, and we'll talk about fermentation later. And this occurs when oxygen is not available, or a lot of bacteria, this is the only way that they can produce energy. And during this, pyruvate is reduced to acetyl-CoA. So pyruvate, when it is oxidized in the presence of oxygen, it occurs in the mitochondria in eukaryotes and occurs at the plasma membrane in prokaryotes. So there are some prokaryotes who don't have mitochondria, and they do this within their plasma membrane. For each 3-carbon pyruvate, during pyruvate oxidation, you get 1 carbon dioxide, 1 NADH, and 1 acetyl-CoA, which is used in the Krebs cycle. The NADH is used in the electron transport chain. So then the acetyl-CoA goes into the Krebs cycle, and it's oxidized. It's an oxidized form of pyruvate. This occurs in the matrix of the mitochondria, and it has 9 steps. We're not going to make you know all the steps, but you should know the 3 main segments. The first one is acetyl-CoA is combined with oxoalloacetate to form citrate. So that happens right here. Oxoalloacetate is a 4-carbon molecule. Combined with acetyl-CoA makes a 6-carbon molecule. The second part is citrate is rearranged and goes through a process of decarboxylation. So it's reduced in carbons, which releases carbon dioxide. This also produces NADH. And the third one is the regeneration of oxoalloacetate. So that 4-carbon molecule is then regenerated to form oxoalloacetate. For each acetyl-CoA, you have 2 molecules of carbon dioxide released. You form 3 NADHs, and there's potential energy there. 1 FADH2, there's some potential energy there, and 1 ATP. Okay, so if you start with 1 molecule of glucose, you get 2 acetyl-CoAs, and this goes through twice, and you would have 4 molecules of CO2, 6 NADHs, 2 FADH2s, and 2 ATPs. Okay, and then you regenerate oxoalloacetate, which is important for that first step. So at this point, you have 1 glucose oxidized to 6 carbon dioxides for waste. You have 4 ATPs, 10 NADHs and 2 FADH2s. So then you have all this potential energy, and you're going to use it to make ATP in the last part, the electron transport chain. This is a series of integral plasma membrane proteins that carry electrons, and as that electron is passed from one to the other, it has energy available from it. It's reduced in its energy state, and that energy can be used for a number of processes. So it's embedded in the inner mitochondrial membrane. It gets electrons from NADH and FADH2. And each complex helps to create a proton gradient to be used by a proton pump, and transfers electrons to the next carrier. So you have NADH dehydrogenase, which uses NADH to have 2 electrons. You have the BC1 complex, which helps pump protons again into the other end of the intermembrane space of the mitochondria. So the final process is chemiosmosis, which we talked about with photosynthesis. You have all these protons on the intermembrane space, and that now makes a concentration gradient where you have a greater concentration on one side than the other. And this membrane is relatively impermeable to these ions, so they can't get through. The only way they can go through is through the ATP synthase pump, which has an enzyme on it which converts ADP to ATP as the proton passes through. Alright, so you put this all together. You have glycolysis, pyruvate oxidation, the Krebs cycle, and the electron transport chain with chemiosmosis, and there you have the process of cellular respiration. The end acceptor on the electron transport chain is oxygen, which is converted into water. So theoretically, you could get 38 ATP for each glucose molecules in bacteria, and 36 for each in eukaryotes. The processes are slightly different. But actual energy is about 30 to 32 per glucose molecule for eukaryotes. And this is because there's a leaky inner membrane, so some of the processes aren't 100% efficient. And the use of the proton gradient for purposes other than ATP synthesis also reduces the amount of ATP you get out of one glucose. So again, oxidation without oxygen can go through anaerobic respiration. Okay, again, these are done by bacteria which live in these really hot and very reactive areas, such as hydrothermal vents and geysers. And many of them use sulfur nitrate or carbon dioxide or even inorganic metals. So some examples of these are methanogens and sulfur bacteria. Or you can go through the process of fermentation. Okay, what happens with fermentation is you go through glycolysis and you get ATP out of it, and you produce NADH. But you need NAD plus in order to complete glycolysis. So in fermentation, it takes an organic molecule and reduces it, and as it does so, it converts NADH back to NAD plus. Okay, and then that NAD plus can be used in glycolysis. In ethanol fermentation, the end product is ethanol, and that happens in a lot of plants mostly. In lactic acid fermentation, which is what most animals do, you have lactic acid or lactate as the end product. So pyruvate as the end product of glycolysis is then converted back to lactate. As it does so, it creates more NAD plus. Alright, so finally, not all energy molecules are going to be glucose. Some other molecules can also be used for energy and inserted into these different parts. So amino acids go a process of deamination. They take off the amino group, and then they can be broken down into different carbon molecules to be used in the Krebs cycle or to be converted into acetyl-CoA. And then fats are similar. They have these long chains of carbons, which can be converted into acetyl groups through a process called beta oxidation. And those, depending on the type of the fatty acid, can then be inserted into, again, the Krebs cycle, or can be converted into acetyl-CoA. So the respiration of a 6-carbon fatty acid yields 20% more energy than a 6-carbon glucose. So it is actually more efficient to store energy as a fatty acid because there's more energy yielding the use of fatty acid than in storing glucose. Alright, so if we'll probably draw something like this similar in class, but this is a whole figure with all our different amino acids and how they are broken down and converted into energy and the byproducts of each. Alright, so that's it.